Hello, everyone. Welcome to my DESeq2 video tutorial for single-cell RNA sequencing data analysis. To identify differentially expressed genes in single-cell RNA sequencing data, we need to create pseudo-bulk RNA data. So in this video tutorial, we will start from a thread object. So for today's analysis, we need to note the library thread, tidyverse, and also we are going to use the scRNA library to compute the size factor for pseudo-bulk RNA sequencing data. And also we know the lemma and the HR, because I'm going to show you two methods to create a pseudo-bulk RNA data from a thread object, including the aggregate expression function from the thread package and the thread to pseudo-bulk RNA function from the HR package. So the thread object I'm going to use for the demonstration is the fibroblast thread object. I prepared this thread object in my pseudo-bulk RNA video tutorial 1 for data preparation. If you want to use this thread object for practice, you can watch my video tutorial for pseudo-bulk RNA video tutorial 1 data preparation. So first, let's load the thread object. You can see we loaded the thread object in the data windows as Fibra. We can use the view function to have a look at the metadata. Let's run. You can see the row names are barcode. We have the origin ident. In this thread object, we have the single-cell RNA sequencing data from 3 healthy control lung and 3 IPF lung. You can see we have the N-count RNA, N-feature RNA thread clusters here, and also we subset the data from the analysis to keep the fibroblast population in this thread object. So all the cell types will be the fibroblast. Then we will use this dataset to perform the DESeq2 analysis to identify differentially expressed genes in fibrotic lung compared to the control healthy lung. So as I mentioned, we have 3 control samples and 3 IPF samples. We can use the unique function to confirm the sample names. We can run. You can see we have control 1, control 2, control 3 from 3 healthy donors, IPF1, IPF2, and IPF3 from 3 fibrotic lung patients. So we loaded the thread object. Next, we can create a count matrix data from the thread object. So from single-cell RNA sequencing data, we create a pseudo-bulk RNA count matrix. So the first function I want to show you is the aggregate expression function from the thread package. You can see the arguments in this function, the object will be the fibroblast object. Then we want to aggregate the count by the samples. We know the sample names are in the origin ident in the metadata. So the group by argument will be the origin ident. We know the matrix data is in the assay RNA and the data is not a count. We can have a look. You can see the assays RNA and the count. So the count is here. We can use the count in the thread object to create a pseudo-bulk RNA. We can set the assays as RNA slot as count. Then we can create the pseudo-bulk RNA for this thread object. let's run the aggregate expression function to create the pseudo bulk RNA data. We named as X. Let's run. You can see we created the pseudo bulk RNA, and we can click this data set. You can see the count matrix is in the RNA, so we can get the count from the RNA, the data frame. We are going to name it as count1 because we are going to show the second function from the HR package. We will create count2 using the scRNA-seq pseudo bulk function from the HR package. So let's get the count1. We can run. We can click the count1. You can see we have 20,680 genes here. The row names are the genes, and we have 6 samples, 3 control and 3 IPF. Here are the pseudo count matrix for each gene in each sample. So you can see we can use the aggregate expression function from the scRNA package to create pseudo bulk RNA data from single-cell RNA sequencing data. So this is the first function. As I showed you in my HR video tutorial, we can also use the scRNA-seq pseudo bulk function from the HR package to create pseudo bulk RNA samples from single-cell RNA sequencing data. We can use this function. You can see the object will be fibroblast, samples is origin ident again, and the class will be cell types. Because in this data set, all the cell types are fibroblasts, so cell types information is not important. So first, this function will create a DG list. We name it as Y for the pseudo bulk samples. Let's run. You can see we have a large DG list. We name it as Y. We can click the DG list. You can see the matrix count is here. Then we can get the matrix count as count2 from the DG list. Let's get it. And we can have a look at the count2 matrix data. You can see the row names are the gene names. We have 6 samples here, control 1, 2, 3, and IPF 1, 2, 3. You can see the column names. At the moment, we included the class information. So we don't need the class information in the column names because the column names are different as our column data. So let's change the column names for the count2. We want to remove the class information. Use the DESeq function. Let's run. You can see now we only have the control 1, control 2, control 3, IPF1, IPF2, and IPF3 as the column names. If we click count1, you can see the gene names are the same, the count matrix are the same. There is a slightly difference for the column names in the count1. You can see the hyphen is not in the bottom. Even we have the hyphen in the bottom in the metadata. You can see the hyphen is in the bottom, but the hyphen is not in the bottom in the count1 matrix when we use the aggregate expression function. So this will have a problem later when we create a DESeq dataset. But we can use this difference as an example to show you we have to get the row names in the column data as the same as the column names in the count data. So I showed you use aggregate expression function or scRNA-seq pseudo bulk function to create a pseudo bulk RNA count matrix from single-cell RNA sequencing data. So we have the counts 1 and the counts 2. Next, we can note the column data. So I have the column data as a text file. We can read in, use the read delim function as the column data. You can see we have the column data. We can click and have a look at the column data. You can see the samples name are in the first column. We have the conditions, donor information, sex information from the donors, and the cell types. As I mentioned, all the cell types are fibroblasts. We want to set the samples name as the row name. So let's use the column to row names function. To set the sample name as row names, let's run. If we have a look at the column data again, you can see now the row names are the samples name. And the row names match to the counter data 2. You can see control 1, control 2, control 3, IPF1, IPF2, IPF3. So the row names in the column data has to be the same name and also the same order in the column names of the count data. So we have the counts and we also have the column data. Next, we can create a DESeq dataset. As I mentioned, the hyphens are different in the count 1 as the code data. We can use this code to check if they are the same or not. If we run, you can see the result is false. So they are different. I showed you already the hyphen is not in the correct position for each sample name. If the names are different, we can try to create the DESeq dataset. Let's run. You can see there is an error because the column names in the count 1 data are different from the row names in the column data. So we can rename the column names in the count 1. Let's run. Now we can create the DESeq dataset again. The count data will be count 1, column data, we loaded the column data. The design will be the conditions. So you know in the column data, the conditions, we want to compare IPF none with control none. So let's try to create the DESeq dataset again. We can run it again. You can see now we can create the DESeq dataset. Let's have a look at the dataset. You can see the class is DESeq dataset. We have 20,680 genes and we have 6 samples. The row names are the gene names and the column names are the names for samples. Under the column data, we have the condition, donor, sex, and the cell type information. As I showed you, we also use the second function from the IGR package to get the counts data from the thread object. We can check if the row names in the column data match to the column names in the counts 2. If we run, I showed you they are the same. If we run this code, we should get a result as true. Let's run. You can see it is true. Again, you can use this code to create the DESeq dataset from count 2. It will create the same DESeq dataset as we created from count 1. Let's run this code. And we got the DESeq dataset. We can have a look at the dataset again. Let's run again. You can see the class is the DESeq dataset. We have the same number of genes and the same number of samples. And the row names are the same as we created the dataset from count 1. And here are the column data and the column data names. So we successfully created the DESeq dataset from a thread object analyzed for single-cell RNA sequencing data. So next step, we can perform the pre-filtering. Because we have 3 control samples, 3 IPF samples, we want to set the smallest group size as 3. Then to filter out the genes that have count below 10. Again, we can click the count data and explain why we set the smallest group size as 3. We can just use count 2 as the example. You can see we have 3 control samples, 3 IPF samples. Because we set the smallest group as 3, and the threshold for the count is 10, so you can see the first gene will be filtered out because all the count is below 10. Then we will keep the second and the third gene. Then after that, we filter out all the genes from here because, for example, this one only 1 sample has the count as 10. So we need at least 3 samples from 6 samples have the count above 10. Then we can keep the gene. So now we can set the smallest group as 3. Then we can set the count threshold as 10. So let's have a look how many genes we will filter out. You can see here we will filter out more than 10,000 genes, and we only keep 9,879 genes for the analysis. So now we can actually do the filtering. You can see before we filter the data, you can see in the data set we still have 20,680 genes. If we do the filtering, you can see we only kept 9,879 genes for the rest analysis. So after the filtering, we can set the reference level for differential analysis. Because we want to identify differentially expressed gene in the IPF lung compared to the control lung samples. So we need to set the levels in this order: control first, IPF second. And also you can use the relevel to name the reference sample as control. Both ways we set the same analysis to compare the IPF samples with the control samples. So let's just use first the code to set the levels. After setting the levels, we can perform the DESeq2 differential expression analysis. So I showed you how to perform the DESeq analysis. For bulk RNA sequencing data, we just need to run the DESeq function without changing any default settings. So the default settings for the DESeq function is for bulk RNA sequencing data analysis. It is not suitable for single-cell RNA sequencing data analysis. So we need to change the default settings here for the DESeq function if we want to perform the pseudo bulk RNA sequencing data analysis. We can have a look at the DESeq2 users manual. You can see here is the recommendations for single-cell data analysis. You can see here the default values for DESeq2 were designed for bulk data analysis, and the default settings will not be appropriate for single-cell data analysis. In the default settings, when we perform DESeq2 analysis, we use the order test, but for the single-cell sequencing data analysis, the recommendation is to use the likelihood ratio test. And also we need to set the use t as 2, minimum as 10 to 6, and the minimum replicate for replace as INF. And also you can see here the default size factor are not optional for single cell count matrix. It recommends use the screen compute sum factors function to compute the size factors. That's why we also noted the screen package in the beginning. To fit the model, we should set the fit type as gamma position generalized linear models. That's the settings for single-cell RNA sequencing data analysis. So we can go back to R to the analysis. So first we can compute the size factors using the compute sum factors function from the screen package. So let's run this code first. Next we can perform the differential expression analysis use the DESeq function. But here we need to set the test as likelihood ratio test. If we use the likelihood ratio test, we need to use the reduced design function. If your samples only have 2 levels, you can use 1. If you have more levels, you need to change the reduced design here. So I'm not going to explain here for more levels. It will depend on your total levels. So you can read the DESeq2 manual to see how to set the reduced design. Then we fit the model as gamma position generalized linear model, and we set the use t as true, and I showed you we need to also set the minimum replicate for replace as INF, and the minimum mu as 10 to 6. So we are going to use those settings to analyze single-cell RNA sequencing data. Now we can run the DESeq function. Let's run. So in the bulk RNA sequencing data analysis, this function is very quick. But for the single-cell RNA sequencing data analysis, we change the default settings. You can see it takes much longer time to run the differential expression analysis. Okay, you can see R finished the DESeq2 analysis for our single-cell RNA sequencing data. Now we can use the result function to get the result for the analysis. We can run. Then we can have a look at the result. You can see down here, the condition is for IPF because we use the reduced design. You can see the row names are the gene names. We have 6 columns from the analysis. The first column is the base mean for each gene. Then the second column is the log2 total changes. You can see we have the p-value and the adjusted p-value in the fifth and the last column. So we can summarize the result. Let's run. You can see we have 241 genes were upregulated in the IPF samples, and 251 genes were downregulated. And also, you can see because this is a single-cell RNA sequencing data, more than 2,000 genes show no counts. So here, the numbers are the upregulation and the downregulation for just the p-value below than 0.1. We can sum the total numbers. You can see we add together, the total is 492 genes. And also you can set the force decay rate as 0.05 to get the result. So let's get the result for the p-value below 0.05. We run the result function again. If we summarize the new result, you can see now we have 147 genes show upregulation, and 132 genes show down-regulation in the IPF fibroblast. If we add the genes together for the adjusted p-value smaller than 0.05, you can see the total numbers now is 279. If you like, you can also order the p-values from the smallest to largest and save it as a result rewarded. Let's reward the p-value. If we have a look at the p-value, order the result, let's run. You can see the gene names were arranged according to the p-value from lowest to highest. We can do more exploration for the result. You can see we can plot the count for individual genes. We know collagen 1A1 is the dominant protein produced by fibroblasts in fibrotic lung. If we plot the count for collagen 1A1, let's plot it. Let's zoom in. You can see in control lung, the count from all three controls are below 1,000, but in IPF patients, you see the count for all three samples are around 5,000. So you can clearly see the upregulation of collagen 1A1 in fibrotic lung. So we finished the analysis. Now we can export the result and save the result. We can save the result as a CSV dataset. If we want to save the original result, we can save the RES. Let's save this one. We can also save the result for the genes rewarded by their p-value. Let's save this one. And if you want, you can also subset the significantly changed genes according to their p-value. For example, here we set as 0.1. If you like, you can also set 0.05 to subset the result. Then we can save the subsetted result as a CSV file. Okay, that's it for the demonstration using DESeq2 to analyze single-cell RNA sequencing data. I want to emphasize that you need to change the default settings for the DESeq function to perform differential analysis for single-cell RNA sequencing data. This is very important for you to get the correct result. I hope my video tutorial could help your data analysis. Please subscribe my channel if you like my video tutorial and share my videos with your friends. Thank you.